Hello dear students. Today we will study about the most important organelle in a cell, which is commonly called as the brain of a cell, and that organelle is the nucleus. The discovery of nucleus was done way back in 1831 by Robert Brown. So let's go into the details of nucleus. Most of the cells have got a single nucleus, and that's why they are called as uninucleate cells. Few cells have many nuclei in them, like our muscle cells, mainly the skeletal muscle cells, and they are called as multinucleate. And there are few cells which do not have any nucleus, like our RBCs. They are enucleate. They do not have nucleus. They lose the nucleus. Also in plants, xylem cells don't have any nucleus. Now let us study about a typical nucleus. If you see in a plant cell and animal cell, you can compare. In a plant cell, the nucleus is shifted to a periphery because of the presence of a large vacuole, whereas the nucleus in an animal cell is more central. Now this nucleus is a double membrane structure. Here you can see the structure of the nucleus in detail. So a typical nucleus have a nuclear membrane or nuclear envelope. And this nuclear envelope is double membrane. You can see there is an outer membrane which is in continuation with endoplasmic reticulum, and there is an inner nuclear membrane, and this nuclear membrane is not continuous. At many places there are gaps, and these gaps are called as nuclear pores. Through these nuclear pores, it keeps on in continuation with the cytoplasm. In the nucleus, there is a process called as transcription. In transcription, DNA produces RNA, mainly mRNA, and these mRNA move from the nucleus into the cytoplasm. That's right, these are the nuclear pores. Okay, and there is a space between them, and that space between the outer nuclear membrane and the inner nuclear membrane is called as perinuclear space. Then there is a substance, a matrix inside the nucleus, that is called as nucleoplasm or karyoplasm. Okay, in the karyoplasm there is nucleus which is without any membrane, which mainly has got protein and RNA in it. Then you can see there is a large network of fiber-like structures which is called as chromatin. This chromatin may be of two types, heterochromatin and euchromatin. Heterochromatin is densely packed and euchromatin is loosely packed. Euchromatin is active transcriptionally, which means only the euchromatin will form RNA by the process of transcription, whereas heterochromatin will not form, that is transcriptionally non-active. Now when a cell undergoes cell division, this whole chromatin, which is present in the form of a network, it undergoes coiling and condensation and forms the chromosomes. Whereas this nucleolus is basically during the cell division, it disintegrates. Okay, its main function is to keep the nucleus in proper order. In a way, it replenishes the substances in the nucleus. So let's now study the different types of chromosomes where this chromatin undergoes the formation of chromosome. they form different types of structures. Let us study what all different types of structures or chromosomes are formed inside a cell when it is undergoing a cell division. So during cell division, the whole chromatin, that is the genetic material, gets coiled and condensed into this structure which is called as a chromosome. Chromosome is actually the unit of heredity. Chromosomes pass on from the parents to the offspring. What are these chromosomes made up of? If you see the structure of chromosome, it has got a structure, central structure, which is called as the centromere or primary constriction. And in the primary constriction, there are small anchor hook-like structures called as kinetochores, which help during the cell division, the movements of the chromosome, which we will study in cell division. Okay, so these kinetochores are there in the centromere. Most of the chromosomes have a single centromere, but few chromosomes have got an additional centromere, which is called as secondary constriction. And after secondary constriction, a small structure is there which is called as satellite DNA. Though satellite DNA does not have protein forming DNA, but it acts as a marker. So in DNA fingerprinting, it is the satellite DNA which is matched. Now, there is, when the cell division takes place, initially the chromosome looks like this, but later on it splits lengthwise. You can see here, it splits lengthwise and remains connected only at the centromere. And these two arms which are produced after splitting, they are called as chromatids. And later on, these chromatids will separate and then it will divide and go into two different cells. This is the reason why the chromosomes get condensed and coiled in a form which can be passed on from one cell to other during the cell division. Okay. So during the cell division, there are different stages which we will study in the next chapter. There is a stage called as anaphase, where these chromatids, they separate and then they acquire different shapes. So based on the shapes they acquire during anaphase, there are different types of chromosomes. Let's now study the different types of chromosomes based on the position of their centromere and what shape they acquire during anaphase. So these are the different types of chromosomes based on the position of their centromere. Now look here. These are metacentric, submetacentric, acrocentric and telocentric. When the centromere is at the center and both the arms, you can see the length of this arm and length of this arm. When the length of both the arms is equal, then we say it is a metacentric chromosome. So what will happen during anaphase? This will separate and this will separate. What will happen? When this is separating and both have got the equal length of the arms, the shape during the anaphase is V-shaped. So we say that metacentric chromosomes during anaphase, they look like V, capital V. Okay, so they are metacentric chromosomes. Now if you see the submetacentric chromosome, in submetacentric chromosome, the centromere has shifted slightly. So as a result, the length of the arms is unequal. You can see this arm is slightly longer than this arm. Okay, so this is called as submetacentric. And during anaphase, if you see, it will give this shape. During anaphase under microscope, it looks like capital L. So, submetacentric chromosomes, they give an appearance of capital L. Fine. Now third, this is acrocentric. In acrocentric, the centromere has shifted more to one side. As a result, one arm is very long and other arm is very short. And under microscope, during anaphase, the chromosome gives this appearance, or you can say appearance of capital J. So we say J-shaped chromosomes are acrocentric chromosomes. And the last type of chromosomes are called telocentric, where the centromere has shifted to only one side. So what happens? There is only one arm. Second arm is not there. So during anaphase, when the chromatids separate, it gives this shape, and which resembles like the shape of capital I. So we say telocentric chromosomes are I-shaped. So just by looking under the microscope, we can make out what type of chromosomes are we dealing with respect to the position of the centromere. This is all about nucleus. Today we have studied about nucleus. Now only few parts are left, like ribosomes, then centriole and flagella, which we will take up in the next session. Okay, thank you.